Hey everyone, we are starting our next unit, Unit C. Specifically, we're starting with C1 regarding cell division. Okay, so from the moment of fertilization, cells are continuously growing, dying, and being replaced. So, two major ways: mitosis and meiosis. Mitosis is where a cell will replicate itself identically. Meiosis is regarding to the production of sex gametes. So they're going to produce an egg or a sperm cell. So meiosis also can be regarded to as oogenesis and spermatogenesis, which was talked about in the reproduction unit. Each new cell or daughter cell, so a daughter cell is a new cell that is produced after meiosis or mitosis, must contain the same genetic information as a parent cell. So this is accomplished through the process of DNA replication and cell division. So looking at the same genetic information, this is relating to mitosis. So this first part is about mitosis, but what we need to look at is that DNA cannot be created from somewhere random, is that it has to come from that original parent cell. So chromosome and cell division. DNA is in the nucleus of each cell. It is tightly packaged into structures called chromosomes. So when we look at a cell, and we know about this going back to grade 10, is that in here we have a nucleus. We have 46 chromosomes in a normal everyday cell, not our sex cells. And that's what we relate to is our chromosomes. And inside the chromosomes is where you're going to find your DNA and that genetic material. So when we look at the DNA, and this is going to relate to the second half of Unit C where we go more into the genetics part, but this is where we have that DNA, which is our double helix. Okay. So if you go and unpackage DNA, it is going to be referred to as chromatin. Okay. Chromatin only condenses into chromosomes when the cell is about to divide. This is important. So the chromatin is going to condense. Each nucleus of a human cell contains about 3 meters of DNA. The other thing we need to look at is when we look at number of chromosomes in a non-sex cell. So I'm not talking about eggs, I'm not talking about sperm, but what I'm talking about are your everyday, your skin, your hair, your nail cells, all will have 46 chromosomes. Now, looking more specifically at a chromosome number, this number of chromosomes will vary from species to species. So some of the examples we have here, like I stated on the previous slide, humans have 46. Okay, and when we say 46, we're relating to non-sex cells. We are looking at the everyday regular cell. Now, these everyday regular cells, these are also called somatic cells. So in case you see that word pop up, a somatic cell is a non-sex cell. Ferns actually have 1,260. A banana has 22. Depending on the species of dog, 78. More chromosomes does not necessarily result in higher intelligence or complexity. So that's important to understand as well, because if we were to compare a human to a fern, yes, a human has less chromosomes, but we are more intelligent as a human species than we are as a fern plant species. The somatic cells of human males have 22 pairs of homologous chromosomes, which also are referred to autosomes, and 1 pair of non-homologous chromosomes, and these are referred to as our sex chromosomes, for a total of 46 chromosomes. So when we look at a picture like this, this is what we refer to as a karyotype. A karyotype is a map of the 23 pairs or 46 chromosomes in a human. So when we look at this, we have them labeled pairs 1 through 22. Okay, those are referred to as our non-sex cells. X and Y here are what is our 23rd pair. And in this case, this individual would identify genetically as a male because they do have that Y chromosomes. So when we hear the word karyotype, they are just taking the DNA and really mapping out the 23 pairs of the chromosomes. When we look more into the last part of this unit, when we talk about genetics, we'll look at some of our common disorders when we look at the karyotypes like Down syndrome, etc., because that is related to having too many chromosomes. There's diseases related to not enough, but that is what we will get a little more into at the end of this unit. So looking over here, some terminology. Homologous chromosomes are chromosomes with the same gene sequence as one another. One homologue is of maternal origin and the other is of paternal origin. What that's saying is you're getting one from mom, one from dad, okay? Because half of the DNA will come from that female or the mom, the other half is going to come from the dad. Although the pairs carry the same set of genes, they may carry different forms of each gene, aka our allele. Because human cells carry 2 pairs of each chromosome, we are considered to be diploid. So when we look at this, the diploid value is represented by 2n. Okay, so that means we have 2. When we look at our sex cells, we are referred to as the sex cells only as haploid. Which relates us to the next here. Human somatic cells are diploid, 2n, and that's how we kind of categorize things. Is that remembering though, is that our somatic cells are our non-sex cells. Human gametes, which are our sex cells, your X's or your X's and Y's, are haploid. And we represent that value by n. So when we look here, 2n equals 46. That is representing the diploid value of a human. This is telling you the total amount of chromosomes. If you were to do algebra, okay, 2n equals 46, isolate the n, I am going to get a haploid value of n equals 23. That value of whatever n is going to give you the chromosome number of the gamete or that sex cell. So in our case, it would be a sperm and an egg. Some organisms, such as earthworms, contain more than 2 homologous chromosomes in each cell. These organisms are considered to be polyploid. So the way we look at this is that we can see something that maybe is a triploid. So 3n equals 54. To figure out what that haploid value would be, we would then solve for n using our basic algebra, divide by 3 here, divide by 3 here. I would get n equals 18. That value for this earthworm is telling me that the haploid is 18. So those are our sex gametes, where a regular somatic cell would have a triploid value, 3n equals 54. Okay, so now that we've kind of covered some basic terminology, looking at DNA, looking at traits, we need to transition to the cell cycle. The cell cycle is your goal is to produce daughter cells with the same DNA as the parent cell. So when we look at this and we're going to start with mitosis, is that we have one cell that is going to then go through mitosis and produce 2 cells. And these 2 cells are going to be the exact same as that parent cell when we look at the genetic aspects of it. Okay, so looking at the cell cycle is that each cell goes through 3 phases before it actually divides. So here we have a cell, okay? Say you fall off your bike, you scrape your knee, you need to produce new skin cells. That one cell is going to produce 2 identical cells. So this is what we're talking about. We're talking about mitosis. This is not sex cell related. This is only producing the same identical daughter cells as the parent. So in order for that mitosis, that splitting of that one cell into 2, is that we need to go through these phases. G1 is where cell growth is going to occur. The S phase is where DNA will replicate, okay, because we need that DNA to replicate in order to have the same amount of DNA in both this cell and this cell. And then we're going to go into the G2 phase, which is going to prep us for division. Collectively, these phases are referred to as interphase. Interphase is followed by cellular division, or M phase. So this is really, really important to understand that this green arrow from here all the way to here is considered interphase. This small section here is where cell division is actually going to occur. So this is where our mitosis, which is our first process, happens. Now, when we look at mitosis, mitosis is performed by all the autosomes. It is a form of cell division that produces daughter cells that are identical to the parent cell. Now, I know there's always the what-if cases. We talk about genetic mutation, etc., but those are kind of your rare cases. When we talk about mitosis producing identical daughter cells, we are talking about what typically happens. Okay, why do we need mitosis? 3 major purposes: growth, maintenance, and repair. Okay, so cute little puppy photo, but same thing. So once this puppy has been born, it's not going to go through meiosis. It's going to go through mitotic growth. It's going to have more skin cells grow, more hair cells grow, more blood cells, etc., until that puppy reaches its full size and capacity. Okay, so few steps in mitosis is that when we look at mitosis, we will look at it in a few number of steps here. So we are starting with, first of all, DNA replication or interphase. So this happens when we go back to that picture of the cell cycle. This is that major piece of that circle, is that DNA is going to replicate. Okay, that those 46 chromosomes are going to double in preparation to head into mitosis. They go through 4 stages of mitosis, but before cytokinesis can occur. So when we look here, we have our prophase. Okay, so what is very important, and the assignment that goes with this topic will relate to pictures and diagrams of cells. So I really hope you guys are watching this video, because I'm telling you, before you start that Google form, it is imperative that you please look at this slide, look at these pictures, listen to my explanations, because it really, really will help you understanding what these pictures are on that Google form. Okay, so the first phase of mitosis, after we go through that phase of interphase, is prophase. Prophase is where each pair of homologous chromosomes have been duplicated. So their cell contains twice as much DNA than normal. Okay, so once again, when you do this assignment that you guys will see on Google Forms after you watch this video, is that it's very important that we pay attention to these details. Okay, so in prophase, chromatin condenses to form chromosomes, nuclear membrane breaks down, and your centrioles move to opposite poles of the cell and spindle fibers begin to form. Okay, so when we look at this, it's prophase is kind of the beginning. That is that your cell is getting ready to go through that phase of splitting. But when we look at these pictures, and it's important that you look at real life cells. So when you see a picture of a cell underneath a microscope during prophase, is that these chromosomes are going to be condensed. So whatever picture I give you, you will notice that the chromosomes are not super clear. They're still condensed inside that cell. There's not a lot of definitions. Chromosomes are not being pulled apart. So that's really important to recognize, is that in that stage of prophase, that we have that condensed version of the chromosomes. Now, when we look at metaphase, metaphase is where spindle fibers attach to the centromere, okay, guiding them to the equator of the cell. So the equator is also the middle of the cell. So duplicated chromosomes line up in single file, forming the metaphase plate. One trick, metaphase starts with M. Chromosomes are going to line up on the middle, so they both will start with that M. Now, when we look at a picture and a diagram, it's really important that we see here, okay, so this is probably an onion tip cell, onion root cell underneath a microscope, but it's very, very important that we notice that those chromosomes are starting to line up in the middle, okay, that they are starting to condense, that they're in that middle on that equator. Now, moving to anaphase. Anaphase is when the spindle fiber shortened and the sister chromatids. So, key word here, you guys, is that our chromosome is that X shape. When we look at a chromatid, is because that X is being pulled away. Is that a chromatid is that chromosome that now has two separate sticks, we can call them. So that X chromosome, which I have here, has been pulled apart into one sister chromatid and another sister chromatid. So this is where these chromosomes are going to be split. Best way to remember it is anaphase starts with an A, away. So when we look at it, these chromosomes are going to be split and they are moving away from each other. So when we look at pictures, same thing. We can see those spindle fibers shortening, okay, these little fibers in here, okay, right in there. You can see that those sister chromatids are physically being pulled away from each other. Now, a little more on this terminology is your homologous pairs versus your sister chromatids. So same thing is that your sister chromatids are those singles. Okay, so when we look at the next phase is your telophase. So chromosomes will fully reach the opposite poles, spindle fibers break down, chromosomes begin to unwind into chromatin, and the nuclear membrane around each will start to form its own genetic information. Now, this one is a little hard to identify, so it's really important that you have a look here. So when we look here, your telophase, if I were to outline these two cells, you will start to see that we are getting close to these cells splitting. Now, it's important to understand that in telophase, these two cells are not split, but you can start to see the beginnings of those two separate cells that are forming. Now, cytokinesis is when the cytoplasm splits to form two identical daughter cells with the same genetic content as the original parent. So when I look here, is the cytokinesis is the physical splitting. They are going to split into two cells. Looking at the diagram, okay, and I know that cells are close to each other, so they're hard to sometimes distinguish if they have been fully split, but there will be enough information given to you guys to show you that it has split. So, on to that point is that telophase and cytokinesis in plant cells look a bit different. So, the one thing that you may just need to be aware of is that because plants, going back to Science 10, contain that cell wall, you will also see that rigid cell wall begin to form. Okay, a little bit more information here. Once again, please look at these pictures. Really, really important to help you in the assignment that I'm giving in this unit. But same thing, prophase. Okay, prophase in this case, sometimes you'll see it as early and late prophase. We will classify it just as prophase. Metaphase, we look here. They're lining up in the middle. Anaphase, okay, they are splitting away. And your telophase, okay, is where they will start to look like they're two separate cells. And cytokinesis is the actual splitting. Okay, this video, I know this is kind of a very new, overwhelming topic. So a lot of these videos have really, really good explanations, really good understanding here to show you an animation of mitosis. Okay, so part 2 of this lesson this week is going to be about meiosis. So the outcome of meiosis is a little different than mitosis in 3 ways. Is that it's going to produce 4 daughter cells rather than 2. Daughter cells have fewer chromosomes than the parent cells, and daughter cells have chromosomes with different combinations of genes compared to those of the parents. Okay, so going back to here for a second. This is why when we start to look at meiosis, we will be producing sperm and egg cells. And what we need to remember is that sperm and eggs are not the same. If you have 30 eggs, those 30 eggs will be genetically different than the rest of them. So this is why, I know minus the twin situation, is that you are different than your siblings. That I am different than my sister, maybe you're different from your brother, whatever the case may be. But this is why genetically, yes, you are related, but you are also genetically different than a sibling. So when we look at this, very good diagram once you get to your Google form to try and identify some of these pictures. But when we look here, we can break meiosis up into 2 sections. Okay, this here is meiosis 1. Here is meiosis 2. So the nice thing is that we will use the same words, but it's very, very, very important that in this phase of meiosis that you need to identify if it's prophase 1, indicating like meiosis 1, or prophase 2, indicating that it's meiosis 2. So please be aware of that, is that the numbers, the Roman numerals 1 and 2, are very, very important in meiosis. Okay, to help us understand before we go into a lot of the details about meiosis, I am just going to take a minute to go through a general understanding of it. So when we look at meiosis, we are starting with 1 cell. This cell is going to be diploid, and it's going to contain 46 chromosomes, and it's also going to be double-stranded. Okay, so when we look at meiosis, meiosis is the production of sex cells. So we are using meiosis, in other words, spermatogenesis or oogenesis, to produce sperm and eggs. That is the goal in meiosis. Okay, so looking over there, keeping in mind that meiosis is going to have a goal of egg and sperm development. So we look here, we have 1 cell. It has it's 2N, 46 chromosomes, and it is double-stranded. So we're talking about, okay, here's our chromosomes inside that nucleus. So please just keep that in mind. Okay, so as we go through here, we have meiosis 1. Meiosis 1 is the first phase, and it's going to go through prophase 1, metaphase 1, anaphase 1, etc., etc. But at the end of meiosis 1, I am going to get 2 cells. Now, we are not done here. Is that we have 2. And what happens is we need meiosis 2 to occur so we can get those 4 daughter cells. Now, this is what's very different than in mitosis, is that mitosis produced 2 cells. Meiosis produced 4. So at the end of the day here, what we have are either 4 sperm or 4 eggs. Okay. Now, when we look at this, what we need to remember is at the end of meiosis is that if we compare it back to the beginning, these are going to each have 23 chromosomes. Okay, they are haploid, and they are also going to be single-stranded. So that is the difference, is that in mitosis, it's producing the exact same thing. In meiosis, we are getting 4 sex gametes, whether it's egg or sperm, that have half of the amount of chromosomes, so they have 23, N for haploid, and they are single-stranded. The reason why they are single-stranded is that when fertilization occurs, and this goes back to the reproduction unit, is that when fertilization occurs, you need 1 egg, 1 sperm, and the combination in fertilization of each will give you a total of 46 chromosomes. Okay, one thing to take note of, you guys, is that with egg production, the one thing is that when eggs are produced, in theory, 4 eggs are produced, and that's kind of all we will go with in Bio 30. But if you read the textbook or if you read any further, is that in reality, that when you get those 4 eggs produced, only 1 out of the 4 are viable at a time. So just wanted to clarify that for you. Okay, on this slide, I just wanted to do a quick comparison for you guys looking at mitosis versus meiosis in a very simple way, but hopefully going into the next set, it'll make a bit more sense. So, mitosis, we are producing the exact same cell. Started with 46, double-stranded, and with 2 cells that are 46 and double-stranded. Okay, so in this process here, is that we are going to go through prophase, metaphase, anaphase, telophase, which will lead us into cytokinesis. In mitosis, we don't use Roman numerals, that they are just prophase, metaphase, anaphase, telophase, cytokinesis, that is it. Now, and yes, I know, I missed interphase. Now, looking here, slightly different when we look at meiosis. Okay, meiosis sex cells start off with 46 double-stranded, 23 double-stranded here, 23 single-stranded, and we get 4. So same thing, wanted to clarify, is that we are going to go through each of the phases, prophase 1, metaphase 1, anaphase 1, telophase 1. Then when I move down to meiosis 2, okay, is that we are going to go through prophase 2, metaphase 2, anaphase 2, and telophase 2, and then we will get into cytokinesis for each of these. So just a comparison, so hopefully moving forward it'll make a little bit more sense. Okay, so going back to meiosis, we'll go through a little bit of these phases a little bit more into detail. So first one is prophase 1. Like mitosis, meiosis begins with interphase or DNA replication. Entering prophase 1, the cell contains 2 pairs of homologous chromosomes. Chromatin condenses to form chromosomes. Spindle fibers form. Nuclear membranes dissolve. Homologous chromosomes align for synapsis or crossing over. Okay, crossing over is the mixing of genes. So this is very important, is that it only happens during prophase 1. So meiosis 1 in prophase, and this is where crossing over of genetic material will occur. This allows each egg and sperm cell to be genetically different than the sperm or egg next to them. So this allows siblings to have those difference in traits.